if problems happen in protein structure, we can monitor this situation using X-ray crystallography experiment, homology modeling, as well as AlphaFold protein structure prediction. AlphaFold analysis is the current approach in predicting, estimating the protein structure. It uses different algorithm, different uh, method compared to homology modeling analysis. Okay. So both homology modeling and AlphaFold analysis, they are computational analysis to predict protein structure, while X-ray crystallography is a type of experimental method. It requires the use of high-end instrument to correctly determine the protein structure. Both approach, experimental and computational approaches, can be used to study the protein structure during disease. Okay. If the disease already established, uh, we we um, we can study the disease development using the same experimental approach, such as SDSBH, SCMS, ELISA, as well as Western blotting experiment. So application, for example, application of Western blotting can be different stages, such as expression level study and also disease development study. Okay, moving on to the first disease that involve abnormal proteins. So it is called cystic fibrosis, which is also caused by a pathogen, namely Pseudomonas aeruginosa. It is related to uh, colonization by Pseudomonas aeruginosa. Okay, in cystic fibrosis, we can see the problems like mutation in a specific protein, namely CFTR protein, cystic fibrosis transmembrane regulator protein. So mutation in this protein or any damage, any defects happen on this protein can lead to cystic fibrosis disease, which is characterized by problematic ion and water movement in lung cells. At the end, we can see a thick and sticky mucus form in the lung and digestive tract, leading to failure of breathing process and others. Other symptoms include, as shown here, diarrhea, chest pain, and others. Okay, cystic fibrosis disease is caused by mutation in CFTR, cystic fibrosis transmembrane conductance regulator. Its name is quite long, so it is the causative factor of cystic fibrosis. Actually, CFTR is expressed and also functional in normal condition. It helps in regulating the flow of ions and water. But when defects or mutation happen on this kind of protein, sticky and thick mucus can develop. So actually, we can see transmembrane domains, ATP, adenosine triphosphate binding cassette domain, and others, they are contained in CFTR protein. We can analyze these kind of domains, transmembrane domains and ATP binding cassette using bioinformatic analysis. Okay, so this is how CFTR protein is localized in our body cells. CFTR protein is completely embedded in the membrane, and it is, however, it is still connected to other proteins and also interacting with other proteins. In normal condition, CFTR protein helps our body cells to pump water and ions. For example, from blood vessel into epithelial cells. Across the epithelial cells, the same goes to ions, sodium and chlorine ions. So the flow of water and ions should be normal when CFTR protein can function. Okay, the mucus is always produced in digestive tract and lung organ because it functions in protecting our organ function. The mucus itself is very important. However, if mutation happens on CFTR, water and ions cannot be transported normally, then sticky and thick mucus develops. So thick mucus, sticky mucus is considered as abnormal, and it can cause other symptoms. So what we can conclude is that if mutation or problems happen in CFTR protein, water movement is inhibited and thick mucus can develop. Okay. Okay, I have one short video. It's not working. Macam mana tadi ni? Okay, I think this short video animation is not working, so we can skip this one. Okay, so we can summarize from a brief introduction to cystic fibrosis. We can summarize that CFTR, cystic fibrosis disease, can make humans susceptible to lung infection. When the mucus is very thick, it facilitates the microbial colonization by Pseudomonas aeruginosa. So this cystic fibrosis disease, it starts with mutation in CFTR, then development of thick mucus, then microbial colonization by Pseudomonas aeruginosa. If no mutation happens in CFTR protein, no thick mucus, and usually no microbial colonization,